So now I'm pleased to introduce our sponsor and one of the moderators for this event, Dr. Steve Brozak. Steve is managing partner and president of WBB Securities, and he will introduce our opening video. Steve.
Hi, I'm Steve Brozak, WBB Research Institute's founder. Since our inception, our mission has been to examine critical real-world issues at the intersection of healthcare and the life sciences industry. In keeping with that mission, we are pleased to offer this video synopsis of the current status and recent advances in CAR T-cell therapy, narrated by Dr. Helen Heslop. The presenter, Dr. Heslop, is a physician, scientist, and a leader in translational research, centering on adaptive immunotherapy with gene-modified effector cells. A professor in the departments of medicine and pediatrics at Baylor College of Medicine, she has authored more than 700 research pieces, and among her honors, Dr. Heslop has been elected to both the Association of American Physicians and the National Academy of Medicine. Now the video.
So therapy of cancer with immune cells should have many potential advantages. These immune cells should be able to have good biodistribution and, unlike monoclonal antibodies or small molecules, should be able to traffic through multiple tissue planes. Immune cells also have multiple effector mechanisms and are self-amplifying and can also recruit other immune effectors. And T cells in particular also have specific receptors that give high targeting ability. This slide shows the receptors that native T cells have that recognize proteins from inside a cell that are presented as peptides by HLA molecules on the surface of the cell. And this is the native T cell receptor, which allows T cells to recognize infected or malignant cells. But recently there's been a lot of interest in an artificial receptor known as a chimeric antigen receptor, and this differs from the native T cell receptor in that it can recognize intact molecules, including non-proteins, on the cell surface. A chimeric antigen receptor is generated by juxtaposing the antigen recognition domain of a monoclonal antibody with signaling components of the T cell receptor. And this resulting artificial receptor can be transferred to a T cell and will give it the ability to recognize tumor antigens on the surface of a tumor cell. And upon recognition, there should be appropriate signaling, resulting in activation and the T cell killing the tumor cell. The initial first generation CARs have been improved by adding additional co-stimulation moieties, most commonly CD28 and 4-1BB, resulting in improved T cell activation and proliferation. Over the last decade, CAR T cells have moved from being a boutique therapy evaluated in phase 1 studies in a small number of centres to being a licensed product. And this is because of very impressive response rates observed in acute lymphoblastic leukemia and CD19 positive lymphoma, as well as myeloma. And one of these such responses is shown here in a patient who had very extensive involvement with a CD19 lymphoma, and after receiving CD19 CAR T cells has a very impressive response. So these are the currently approved therapies as of August 2023. There are 4 therapies targeting CD19 in a variety of CD19 positive malignancies, and 2 therapies targeting BCMA in myeloma. The current commercial CAR T products are living drugs which have significant side effects. So a certain infrastructure is required to access this therapy. The patients have to be able to be apheresed at an apheresis centre at a tertiary hospital that requires a local cell processing facility to assist with handling. The centres that administer these cells need contracts with pharma companies, agreement with insurers, and expertise in handling complications, such that most companies require such centres to be FACT accredited for immune effector cell therapy. So while these CAR T cells can have very beneficial therapeutic responses, as I showed in the previous slide, not all patients respond, and even patients who do respond can eventually progress. So there are three major causes of failure. The first is tumor-specific and includes loss of the target antigen in many cases. The tumor microenvironment can also impede the activity of the infused CAR T cells, and sometimes the infused cell product may not be optimal because of the patient's previous therapies. So future directions are aimed at improving activity, and there are several ways to do this. One is to use CAR T cells earlier in the course of the disease, and while initial approvals were for relapsed or refractory disease, 2 of the products are now approved as second-line therapy. Another approach is to target multiple antigens so as to reduce the risk of tumor evasion due to downregulation of one antigen. Another area of interest is in simplifying the logistics of manufacture for an autologous product. And several academic centres and companies have now developed CAR T cell products where the manufacturing time has been reduced to 2 to 3 days. And these products are now being tested in clinical trials. Another strategy is point-of-care manufacturing, which is being explored in several European centres where the products are manufactured locally, as we do for standard of care bone marrow transplants. Another option is to try and overcome some of the limitations of an autologous product by using banked allogeneic CAR T cells. So the limitations of an autologous product are that it's patient-specific and it may have variable quality if the patient has been heavily pre-treated. As I've discussed previously, it's time-consuming and expensive. So another option is to make banked cells from a healthy donor, where you'll have an allogeneic off-the-shelf, or more accurately out of the freezer product, that will have predictable functionality and be readily available and hopefully eventually also have a lower cost of goods. So as shown here, instead of having a manufacturing process which involves ex vivo expansion and quality testing and may take 28 days from an autologous donor, you can have a product immediately available that is being manufactured from an allogeneic donor. Another area of current research is trying to extend the success that we've seen in CD19 and BCMA positive cancers to other hematologic malignancies. CD30 CARs have shown activity in Hodgkin lymphoma, and there are multiple targets that groups are exploring for AML and T-cell lymphomas, but the challenge with these tumours is the same antigens are also expressed on normal cells. And while it's acceptable to deplete your normal B cells with a CD19 CAR, depleting normal myeloid or T cells has more serious consequences. So at present, using CAR T cells for these 2 malignancies is a transplant-enabling procedure where the patient is subsequently rescued with an allogeneic transplant. Another challenge is extending this therapeutic modality to solid tumours. Solid tumours present much more of a challenge because they have a number of other infiltrating cells and a tumour microenvironment that's hostile to adoptively transferred T cells. So there's a number of strategies that can be explored to try and overcome these tumour evasion mechanisms. And these include modifying the CAR T cell with additional moieties, so you also inhibit agents such as TGF-beta produced by the tumour. You can also try and optimize trafficking to the tumour site, and there's a number of genetic modifications that are being tested in the clinic to enhance signal 3 so that T cells have better long-term persistence. So the current state of the art for CAR T cell therapy is that up to 50% of patients with refractory B cell malignancies who had very few available previous therapy are now showing durable, complete responses to CD19 CAR T cell therapy. In addition, over 50% of myeloma patients who receive CAR T cells respond. And the major research focuses are trying to extend this success to other malignancies, to overcome tumour evasion mechanisms, and to simplify production and increase accessibility of this therapy.